Thanks everyone for joining today and welcome to the GA4GH 101 webinar. So GA4GH stands for the Global Alliance for Genomics and Health, the international standards body for genomics. We are members of the GA4GH Secretariat team. I am Justina Chung. I'm the operations lead. I'm based out of the Ontario Institute for Cancer Research in Canada. I'm joined by Julia. Julia, do you want to give a brief introduction as well?
Yes, I'm Julia Ostman. I am the GA4GH science writer, and I'm based in the Boston area at the Broad Institute of MIT and Harvard.
Thanks. So Julia and I will be giving the presentation today, but I also want to give a shout out to Nirja and Stephanie, who are also here helping us run the webinar, and you'll see them come up and chat and can answer any questions, or if you have any technical difficulties with Zoom, they can help with that as well through chat. And so we're excited to have you all here. In this webinar, we will explain what GA4GH is, why the standards it creates matters, and how you can get involved. So today we'll be using Slido for participation and Q&A. We really do want this to be an interactive experience for everyone. If at any point you have any questions, please feel free to raise your hand using the raise hand function in Zoom. But you can also use Slido either at the slide.do link. You can use hashtag GA4GH101. You also can use your mobile phone to scan the QR code on the screen right now if you like to use your mobile phone to input your questions. And then I do want to make a note that Julia and I are non-technical staff members here at GA4GH, so you're very welcome to send in any technical questions. However, if we aren't able to answer them, we'll happily connect you with the right members of the Secretariat team and GA4GH community to help answer them. All right, so to get used to Slido, we'll kick things off with a couple of polls. If you want to just go ahead and join Slido now, there should be an active poll for you to participate in. And the question here we have is, where are you joining from? Toronto. Nice. That's where I am as well. And we'll just give it maybe a minute here so people can type in their answers. Somerville. Welcome.
I see Boston and Mexico in the chat. Welcome.
Welcome. Seattle, Australia, Indiana, San Diego, Edmonton. Taipei. More Toronto. Montreal. This is great. Washington. So welcome. It's nice to see everyone from all over the world. So we got 11 answers in. That's great. I'll move on to the next question. What's one thing you look forward to learning today at this webinar? I see lots of typing. How GA4GH works. We'll definitely cover that. What GA4GH does and the standards. Standards, yes, we will definitely cover that today. What's next for GA4GH? Overview and status. How GA4GH can, what GA4GH can do to help standardize phenotype terms. How information can be shared. All right, this is great. I'm glad everyone's getting comfortable now with Slido. And I'm happy to share that we will be able to cover most of these, so please look forward to that. Last question is, do you currently engage with GA4GH? And if so, in what ways? If you use one or more GA4GH standards already, if you work with one of our driver projects, if you've attended any of our plenary meetings, if you're part of any of our work streams, and if this is all new to you, no worries, we will cover all of this. And this is a multi-select option as well, so you can choose more than one. You can choose all that apply. Great. So it looks like a majority, or almost half of the respondents aren't currently engaged. Great. So hopefully by the end of this webinar, we can help you learn more, and you'll learn how you can engage with us. All right, so Slido will be available throughout this presentation. You can type in your questions there if you don't feel comfortable raising your hand or using the chat function. And then we'll have a little pause to answer some questions, and then a full Q&A session at the very end to pull up all the questions that are still unanswered. And so let's dive right in. This is how the webinar will run. We will go through why GA4GH exists, how GA4GH works, the future of data sharing, how GA4GH can help you and how you can get involved, and then some time at the end for the Q&A. So now I'll pass it on to Julia.
Thank you, Justina. So in terms of explaining why GA4GH exists, so I think many of you on this call are aware that genomics has seen massive and ongoing changes in technology. And just one basic example, sequencing the human genome, what cost $3 billion and took a decade, over 20 years ago, can now be done for less than $1,000 and take less than a day. And this creates its own special set of challenges. So next slide, please. One of those challenges is scale. Here is a study from last year of an analysis of more than 450,000 participants from the UK Biobank. And on the next slide, we'll see that it's not just number of participants. There are more than 200 genomic data initiatives globally. And this is in the clinical setting, in the laboratory, on the national scale, and in cohorts of participants in clinical trials and other places. And on the next slide, we'll see that these more than 200 initiatives are generating huge amounts of genomic and related health data. And we at GA4GH are seeing a shift. That data is increasingly coming from clinical care and clinical trials rather than from academic research. Next slide, please. And that leads us to yet another challenge, which is creating this pipeline between research labs and the clinic. So here are two recent UK government policy papers arguing that this effort to build the pipeline is absolutely crucial, that it saves lives. So now we're going to do one more poll, and we're going to ask you, what is the biggest problem that you face in data sharing and/or precision medicine? So feel free to write in, what is the biggest problem that you are facing? Lack of access to relevant resources, governance. Yeah, those are two big ones. Lack of standards and implementation guidelines, accessing variant information, unstructured clinical data. Yeah, I definitely think we're going to cover some of this today, and is certainly the work that GA4GH does, moving and storing large amounts of data. Absolutely. I think a lot of people would relate to that one. Great. Well, it seems like folks have stopped typing, so thank you for sharing these problems, and we hope to address some of them some of them today. Oh, we've got two more. We'll wait for them and then I'll hand it back to you, Justina. Okay, I think we're good to go. Over to you.
Oops, sorry, I was muted. Thanks, Julia, and thanks everyone who participated in that last poll or survey. So now we'll tell you a little bit about how GA4GH tackles these challenges in genomics, health, and data sharing. The GA4GH mission aims to accelerate progress in genomic science and human health by developing standards and framing policy for responsible genomic and health-related data sharing. And GA4GH was founded in 2013. Here is our current leadership. We have 4 members of our executive committee, which include Ewan Burney, who is based at EBI in the UK. He is our chair. Peter Goodhand is our CEO, based at Ontario Institute for Cancer Research in Canada. Heidi Ream, who is our vice chair, based at the Broad Institute in the US. And Catherine North, who is also vice chair and based at the Murdoch Children's Research Institute in Australia. And then you see at the bottom half of the slide, we have our steering committee. It's our leads of our work stream and champions of our driver projects make up our steering committee. And we will go over who or what the work streams and driver projects are in just a little bit. So what can the sharing of global genomic data do? If we can create a virtual cohort of properly consented global genomic data, we'll enable a host of downstream impacts for research and precision medicine. We'll be able to demonstrate patterns in health and disease, increase statistical significance of analyses, lead to stronger variant interpretations, increase accurate diagnosis, and advance precision medicine. But data is all over the world. To answer the most pressing research questions, we need interoperability between computer systems and platforms, between data pipelines and databases, between many different jurisdictions. Standards are what create this interoperability. So at GA4GH, we build interoperable standards by convening a vast community at meetings and across our ecosystems. And this diagram here summarizes this GA4GH ecosystem and its outputs. So we have over 670 organizational members who have committed to the mission of GA4GH. We have representatives from more than 90 countries. More than 450 people contribute actively to developing our standards. We serve as the clearinghouse of information and knowledge throughout this community, and we have over 4,000 people subscribed to our mailing list currently to receive updates from us. And we'll explain shortly the actual work of our standards creation through our 8 work streams and how we work closely with 24 driver projects and other initiatives around the world. And all these people working together has produced more than 15 regulatory policies and frameworks, and 23 technical standards. We have seen this in over 60 publications and implemented more than 120 times around the world. All this is how we help enable a healthcare system that learns from global knowledge. And now we'll take a closer look at how GA4GH works. So I had mentioned we have our 8 work streams. I won't list them all now. We will dive deeper into each one later on. These are internal GA4GH groups where the standards are actually developed. 2 community members volunteer to lead each work stream, and contributors from all over the world join these work streams to contribute to these standards and attend our meetings. 2 of these work streams are foundational, that is the regulatory and ethics work stream and the data security work stream. And the other 6 in blue are what we call technical work streams. And so contributors of these 8 work streams collaborate very closely with 24 key driver projects, which you see on the right hand of the slide. Out of all the genomic initiatives in the world, these 24 projects have committed to giving resources and insight into our work streams and to pilot new standards in real world situations. So some examples are Autism Speaks, Matchmaker Exchange, Genomics England. These driver projects cover all sorts of areas from complex traits, rare disease to cancer, And then on top, you see the map, which reflects the geographic spread of our driver projects. And it really represents our strategy to ensuring we meet the needs of as far-reaching a community as we can. And I'll pass it back to Julia.
Yes, thank you. So here is the opportunity that GA4GH seeks to take. The pandemic slowed us down a little bit in terms of genomic data sharing, but we also learned a lot about setting up fast-moving international collaborations. So can we leverage the pandemic emphasis on infectious disease data sharing and enable the secondary use of clinical genomic data for research? If we can do that, we will fast approach a virtual cohort of 60 million samples. Next slide, please. And here at GA4GH, we've identified 4 different approaches to sharing that level of data. One is through a centralized genomic knowledge base, where you can access the knowledge that has been developed from data, but you can't access the data itself. Another way is through a data commons or a trusted repository of multiple data sets that are all held under the same sharing agreements. And then there's through some kind of distributed system where the data is hosted locally, but analyzed remotely. And so one example is through the hub and spoke model of federation, which requires a common agreement and shared technological standards. And another example is linking up disparate data sets all over the world. And again, this requires common technological standards. So we're going to jump to another poll. Which data sharing approaches have you used among these 4? So the centralized genomic knowledge bases, the data commons, hub and spoke federation, or the linking up of distributed data sets around the world. And you can choose more than one on this poll. We'll give a couple of minutes to read through, make some choices. Okay, so far we're seeing data commons, and data commons is still the winner, followed by centralized genomic knowledge bases. Okay, this is great. So it's great to see that there's at least someone on this call who's used each of these 4 approaches. Oh, I see centralized genomic knowledge bases and data commons are in a tie. This is very useful. Great. Well, we will definitely talk more about these different data sharing approaches, but it's helpful to see where everyone is at right now. All right, shall we jump to the next slide? So something that we have noticed at GA4GH that affects all data sharing approaches is a shift in the number of genomes being stored in the cloud versus in on-premises databases, for example, like an institutional server. And you can see an estimated range on this slide for that increase in genomes in the cloud. Next slide. Another shift that we're beginning to see is from data copying to data visiting. So instead of downloading multiple copies of a large data set to individual local machines, I think somebody mentioned that as their biggest problem right now in data sharing, now researchers can send virtual machines to visit the data where it lives and perform analyses remotely via standardized APIs. Next slide. And we think that this model of federation, and it comes in different flavors, including some of the data sharing approaches we described earlier, will likely dominate the genomic data sharing landscape going forward. And when it does, more research questions will open up simply by the fact that more data is available to be visited. So federation, just to get into it, allows data to remain in its location of origin. And it provides broad access methods which respect the regulatory processes of that region. So sometimes this is referred to as bringing the code to the data. A federated system between healthcare and research will also allow us to transfer methods and skills into the healthcare sector. And it will let us use that healthcare data for secondary research to make more discoveries about humans. Next slide. So if we do manage to create that feedback loop between healthcare and research, if we can continuously feed data and lessons between the two, then we will have achieved what we call the global learning health system. And this diagram, which I'll get to in a second, is one example of a learning health system. But that term is used to describe a continuously adapting system in which data and analytics generate knowledge, and then that knowledge is implemented into practice, which changes behavior, and that creates new and better data, and we feed it back into the system, ultimately to generate new knowledge. So it creates this kind of ongoing virtuous cycle. So in this example of the learning health system on the screen, it's a hypothesized data life cycle. So we can start on the left with that data donor. It begins with obtaining consent from them, then moving through to responsible data storage and also access. And then finally, it supports secondary research within a federated network, like I was describing. And all of that leads to improved knowledge and informing health and medicine. So when we go to the next slide, we can see that the standards GA4GH produces, and you can see them here, all these little standards next to the GA4GH colorful logo. These standards play an integral role in each step of a data lifecycle and an overall learning health system. And it's not just this one hypothesized cycle. These GA4GH resources are extremely flexible. They can be adapted to other contexts and systems and work together in different ways. And to tell you a little bit more about how this all works, we are going to play a brief video.
University Hospital is a global leader in cancer research. Anna is a member of the UH faculty, where she leads a team in studying a deadly pathogenic disease. Like many of her colleagues, Anna would like to work with more datasets around the world in order to analyze the genomic and health-related data of as many patients as she can, in order to identify genetic variants associated with better outcomes and guide better clinical treatments. Similarly, UH and institutions like it want to make their troves of data available to researchers like Anna. But without interoperable methods for researchers to efficiently identify, access, and analyze data of interest to them, none of this is possible. Right now, Anna's team can easily interrogate local UH data, but working with datasets around the world is challenging. Without large interoperable cohorts, the statistical significance of their results remains low, limiting their ability to advance the field. But it doesn't have to be this way. A suite of software tools powered by GA4GH standards can connect researchers like Anna to a global network of data in a secure and responsible way, unlocking its potential to benefit human health. First, the GA4GH Discovery APIs allow data stewards to make their datasets findable. Anna can query this large network to identify datasets that are relevant to her study. For each dataset she wants to analyze, Anna works with the corresponding data access committee to acquire permission. GA4GH standards help these groups to reliably determine whether Anna's proposal is within the scope originally agreed to by the data donors. Her approved requests are added to her virtual identity. And now she can begin working with the data. With the GA4GH Cloud APIs, Anna's lab is transformed into a virtual hub, connecting her to all the data she needs to do her job well, although she will instruct software machines to carry out much of that work on her behalf. Anna tells each machine where to go, what data to analyze, and which analysis tool to use. Equipped with these instructions, the software machines head out to analyze a portion of the data right where it sits. Only machines with authorized access to this data are allowed to enter. The machines carry out Anna's instructions, which is made possible because the institution has packaged its data in a universal way, also according to GA4GH Cloud standards. Each machine brings back its analysis result to feed into Anna's study. As a security measure, the data steward may remove access from Anna's software machines once the analysis is complete. Anna's team fits the pieces together to reveal a genetic variant associated with better outcomes, a new insight they could never have seen if the datasets were not interoperable. Anna is just one researcher, but her story isn't unique. Someday, we hope that every researcher in the world can access a global network of data from the comfort of their lab, revealing otherwise hidden knowledge of human health and disease. University Hospital.
Thank you so much for watching that. Hope you enjoyed it. So we're going to jump into one last poll, sort of based on the presentation so far. We're curious to ask you, which data sharing approaches do you want to try? And again, you can pick more than one going forward in your work. We'll give about a minute for folks to answer. So we're seeing hub and spoke, a good deal of interest in hub and spoke federation, still data commons, and about equally so far centralized genomic knowledge bases and linkage, more on linkage of distributed and disparate data sets. Great. Well, thank you so much for taking the poll. And now we're going to pause briefly for about 5 minutes to answer any questions that you may have already submitted, or if you have any questions up to this point in the webinar, please feel free to submit them here on Slido, or you can put them in the chat, or you can raise your hand if you would like to say them out loud. And we will have more time for Q&A at the end of the hour, but we will just pause briefly if anyone has questions now. Okay, so far not seeing any questions. We'll give 30 more seconds, and otherwise we will move along to how GA4GH can help you. Okay, we've got a question. So a question has come in, does GA4GH look at the sharing of clinical data for clinical purposes in the healthcare environment? Yes, the answer is yes. I'll take a stab at this, and then Justina, if you want to jump in. So we actually just last week, the regulatory and ethics work stream, which we'll talk about in a little bit, had a new standard approved, the clinical genomics consent clauses. And this standard is sort of a menu of phrases and clauses that clinicians can use in a clinical setting to create a consent form for different kinds of, in this case, it was genomic testing, specifically for use in the healthcare environment. It would be a different consent if it were going to secondary research. Justina, can you think of any other examples?
No, that was a great example.
Great. And if you want to, we'll have lots of contact information later, but if you want to reach out to us with your email, we can put you in touch with those REWS colleagues who I'm sure will have a lot more to say. Okay, we have another question. I like the little analysis robots in the video. In practice, how would they be implemented? So there are several ways in which the whatever you would like to call them, virtual machines or software machines or bots would be implemented. The primary method is by using GA4GH standards in order to make sure that they could travel seamlessly between different databases, different environments. But I'll give one example of a recent usage of this federated analysis, or moving the software to the data. So a group based at UC Santa Cruz put together a Docker software container and sent a series of analysis tools to some colleagues at Riken, one of the Riken centers, and it queried some data related to breast cancer. And now that Docker, sorry, I should finish the story. And that once it queried the data, it was able to send de-identified summary results to colleagues in Australia who were able to create some stronger variant interpretations for breast cancer. But that Docker software, that container, is how the analysis was able to travel to the data. And it is now available on several platforms that use GA4GH standards that we'll talk about later, such as REWS, the Workflow Execution Service. And again, if you want a more technical answer than that, please email us and we'll get you to the experts.
And I would just add, right now, implementation is a big focus at GA4GH, and we'll get into it later, but we do have an implementation forum that looks specifically at how GA4GH standards can be implemented in the real world, how projects can pilot them. So I can get into that later. Went for it too much. So now let's get into how we can help you and ways that you can get involved. So we'll be moving into a deeper dive discussion on each of the work streams we previously mentioned, including opportunities for you to get involved and help produce and maintain standards that will benefit your work. And so Julie and I will alternate and go through each work stream. I'll start with the clinical and phenotypic data capture, or ClinPheno work stream for short. So ClinPheno supports clinical adoption of genomics by developing information models and standards for describing and exchanging clinical phenotypes in genomic medicine and research. This includes the capture and exchange of information between clinical, research, and patient-centered systems. There are two main focus areas in ClinPheno. One of them is PhenoPackets. It's a standard for phenotypic information. The standard aims to facilitate communication between the research and clinical genomics communities by creating an ecosystem of interoperable tools and resources that can use phenotypic data with fewer barriers. The second standard that's developed by ClinPheno is the pedigree standard. It's intended to allow for the computable exchange of family health history, as well as representation of larger, more complex families. It is a graph-based model intended to fit within the structure of other standards, such as HL7 of FHIR or PhenoPackets. The pedigree team right now is actually welcoming feedback from the community on the standard and associated deliverables at GitHub. And Neerja and Stephanie, maybe if you can help paste that GitHub link, anyone who is interested in reviewing the standard is welcome to follow that link and provide their comments. A third emerging focus area within ClinPheno is computable cohort representation. This group is right now exploring the space of cohort discovery, extraction, and comparative statistics, and are in the earliest stages right now to defining potential standards that can come out of this group. And if you have any interest in the three focus areas I've mentioned, or if you have any questions and would like to get involved, the contact is Lindsay Smith. She is our workstream manager, and you can find her email address on the slide.
Great, so I'm going to jump in with the Regulatory and Ethics Workstream, or REWS. I mentioned this briefly just earlier, but this workstream is built on a novel human rights framework, and it seeks to create and harmonize forward-looking consent and privacy policies, as well as anticipatory data governance models. So its goal is really to activate the human right to benefit from science. And this work stream does it with a focus on ethical, legal, and social implications of international data sharing. So it puts out many different kinds of deliverables. We don't have time to go over everyone today, but to highlight just a few. So I mentioned the clinical genomics consent clauses earlier. There is in fact an entire toolkit of consent clauses available for different kinds of data collection purposes. And it is a menu of options for researchers and or clinicians when they are creating consent forms. Another example of a deliverable for REWS is the engagement framework, which has tips for engaging with participants, patients, and the public. And then the Data Access Committee Review Standards, or DACRIS policy, helps data access committees consider requests to look at the data that they have. And there are many ways to get involved in REWS. If you're at all curious, there are 5 new subgroups that are coming up. So if you're interested, maybe screenshot this slide. There's one on diversity in data sets, ethical provenance toolkit, public attitudes for genomic policy, which is built on a landmark survey called Your DNA, Your Say, clinical data sharing and consent, and newborn genomic sequencing. And if you at your institution have a data access agreement that you're able to share, that new ethical provenance group is looking for such agreements to compare and find common causes and similarities. So contact Lindsay as well at the email address on the screen if you can share that.
Great. We'll move on to the Data Use and Researcher Identities Workstream, or DURI for short. DURI aims to enable researchers to efficiently discover genomics data and then apply to get access automatically based on their digital identity, and if it's required, based on a machine-readable research purpose to access datasets with use restrictions. The 3 main DURI standards include GA4GH passports. It's a standards-based digital identity that researchers can use to access distributed data. This includes ways to express data access rights. The passport contains visas as evidence of past access permissions that can be carried across institutions and data environments. Version 1.2 of passports is currently open for community input, and you can share your comments at the GitHub link, which is now shared in chat. Thank you. The second standard by DURI is the Data Use Ontology, or DUO. It allows data custodians to semantically tag data sets with restrictions about their usage, making it possible to automatically discover data sets based on the authorization level of users or intended usage. And then lastly, the machine readable consent guidance was developed by DURI in collaboration with the regulatory and ethics workstream that Julia just described earlier. The consent guidance describes how to adopt data sharing consent language that unambiguously maps to DUO. This facilitates data access request submissions and approvals.
Next up is the data security workstream, which helps the community to keep data protected and ensure that GA4GH standards are developed within a sound risk management framework. And so the deliverables for this workstream include a risk assessment methodology for software stacks, which includes defining what information and data needs to be protected, as well as guidelines and recommendations for cloud security and privacy. And then the Authorization and Authentication Infrastructure Standard, or AAI, which is the technical profile that underlies the passport standard that Justina just mentioned. This workstream is looking for volunteers to lead 2 newly forming groups, the breach response policy and malfeasance rules. And if you have used malfeasance rules in the past or know of a really great use case, they would love to see it. So please contact Fabio Liberanti. He is the workstream manager for data security.
Moving on to the discovery workstream. The discovery workstream develops standard APIs and context-aware user interfaces, aiming to create a unified data discovery platform that makes it easier to find and use data, as well as tools and infrastructure for genomic and clinical analysis. So standards developed by this work stream include the Beacon API. It's a federated web accessible service that can be queried for information about a specific genomic variant and then returns a report on its existence in the queried resource. Another standard developed by this workstream is Data Connect API, which aims to address the challenge in publishing and discovering biomedical data, which is often stored in a variety of locations, methods, formats, and file types. Data Connect consists of a common JSON schema, which data providers can use to describe their data without complete transformation and harmonization into a new model. Data Connect also consists of a method for searching data using custom queries. Right now, the team is looking for groups to test and implement the API. The discovery team is also interested in working with others on ways of describing shared data with uses relating to both Data Connect and Beacon. So if you are interested in exploring this, please contact Fabio, who is also the workstream manager of Discovery. And then lastly, some other standards developed by Discovery include Service Registry and Service Info. Both together provide a network infrastructure for all API-based GA4GH standards to create an internet of genomics.
Great, moving on to the Genomic Knowledge Standards, or GKS workstream. The goal of this workstream is to enable downstream analysis of genomic data by developing common APIs for exchanging reference genomic information. So the aim here is really to develop some consistency in exchanging information related to genomic sequence, annotation, as well as associated metadata and provenance. So some standards developed by GKS include the Variant Representation Specification, or VERS, which is a standardized schema representation of many different forms of genetic variation. And if you're curious to learn more about this or adopt it in your work, and you happen to be going to the ISMB conference next month in Madison, Wisconsin, you can sign up at this link here for the VERS hackathon that we are hosting. Another deliverable for GKS is the Variant Annotation Specification, or VA, which is a standardized schema that focuses on interpreting observed variants with details about how they may functionally impact a gene function, so leading to progression of a disease or how effective a particular therapeutic may be given a variation profile. And there's also a newly forming GKS group, a sequence annotations API group, that is looking at discontinuous and continuous annotations on GA4GH genomic sequences. And there is one more opportunity to meet the GKS crew if you will be in Switzerland next month at the International Genome Graph Symposium. There will be some GKS and GA4GH representation on the program, which you can see both of these in the links in chat. Thank you. And for more information, contact the workstream manager, Reagan Thomas.
Great. Moving on to large-scale genomics, or LSG for short. The LSG workstream develops efficient formats to store, access, and analyze sequencing reads, genetic variation, and gene expression information. Some file format standards that are maintained by LSG include the Sequence Alignment Map, or SAM format. The team also maintains and develops its binary counterpart, BAM, as well as CRAM, the format for long-term compression of genomic alignments. LSG also maintains the variant call format, VCF. VCF version 4.4 is currently open for comments in GitHub. GHIF4GH is a standard file container format, which is also being maintained by LSG. It allows genomic data to remain secure throughout their lifetime, from initial sequencing to sharing with professionals at external organizations. And right now, the team is working on a landscape analysis for the development of the GWAS data formats. So if you're looking to get involved with that, please contact Reagan Thomas as well, who is the workstream manager for LSG.
Last but certainly not least, we have the cloud work stream. So modern genomics involves running computational workflows over long periods of time to transform and derive insights from large data sets. And the cloud work stream really wants to help the genomics and health communities take full advantage of modern cloud environments by bringing algorithms to the data, as we've discussed before. And so the solution that the work stream has come up with is releasing a suite of 4 API specifications, which allow for cloud-based genomic analysis that is platform agnostic, which is, it is not tied to any particular cloud provider or infrastructure. And these 4 APIs are the Tool Registry Service, or TERS, which provides a standardized interface for accessing published workflow algorithms. Then there's DURS, Data Repository Service, which allows clients to choose which copy of a data set to submit to a workflow. And then finally, the Workflow Execution Service, WES, and the Task Execution Service, TESS, act as interfaces for clients to request that a workflow be run on a data set of interest. And the cloud work stream is gearing up to update these 4 APIs and expand the team. So if you're at all interested in getting involved, please reach out to Reagan.
So aside from the work streams where standards are developed, we have other groups that are focused on testing out existing standards to see how they can be put into practice to facilitate real-world federated genomics. So first we have the Federated Analysis Systems Project, or FASP. This was established to show that GA4GH APIs, when used together, can facilitate real-world science by enabling genomic data discovery, access, and analysis in the cloud. FAST conceptualizes demo use cases that simulate how a researcher would search and analyze genomic data within the GA4GH ecosystem. We also have the GA4GH Starter Kit. It's a suite of open source reference implementations. Each application acts as a server implementation of a single GA4GH API specification. So this was developed primarily by the staff engineering team, and the Starter Kit was designed to be deployable in a variety of contexts. So the Starter Kit will serve an important role in the improvement of our standards by providing a code base that is easy to update with prototype features. And as new specification features move from experimental to standardized, the Starter Kit will keep up with these developments so that there are always reference implementations of the latest standards. I mentioned earlier, we have what we like to call an implementation arm of GA4GH, which is the Genomics and Health Implementation Forum, GIF for short. The GIF's overall goal is to support the adoption of GA4GH standards and provide a mechanism for implementers to provide feedback back into the development teams. It will ensure that the standards fits the need across multiple initiatives and healthcare systems, and it encourages diverse input and the sharing of challenges, best practices, and opportunities. So right now, all the icons you're seeing are the current listing of GIF members. You can learn more about GIF on our website under the implementation tab of our navigation bar. And you can also learn there how you can be added to this list of initiatives. So we know that was a lot of information. If you didn't catch some of the contact information, you can also email the Secretariat team at secretariat@ga4gh.org, and we'll make sure to put you in touch with the right person. We will also share these slides with everyone later on as well, in case you didn't get a chance to screen cap any of the slides. We do welcome organizations to join us as an organizational member. You can apply at ga4gh.org/members. You can also sign up for any of our mailing lists to receive updates on our work, upcoming meetings, and more at ga4gh.org/subscribe. And if any of the work streams are of interest to you, we have a great program to help you get started. The GA4GH onboarding program was introduced just last year and connects newcomers with workstream guides to help orient and welcome them to the GA4GH community. if you are interested in joining a work stream and being part of this program, you can contact Neerja Skantharaja to sign up. I have her email address on the slide here, but she is also here on this webinar, so you can also send her a chat if you'd like. So yes, it's been a very successful program so far, and so I highly encourage you join if joining a work stream is something you are looking to do. All right, so this brings us towards almost the end. As promised, we have our Q&A session to answer any questions you may have. So again, you can do this through Slido, through the chat, or the raise hand function in Zoom. Oh, sorry. Let me. So we already have one question here. How are these standards versioned, and how is the support for moving people from one version to another, and what hindrances does GA4GH see regarding that? So should I take a stab at this first, Julia?
Yeah, go.
So we do have what we call a product approval process that standards go through whenever a new version or a new standard is put out. And I believe we have that process kind of outlined in our website. But as standards evolve, and new versions come out, whenever there is a breaking change to a standard, it does have to go through the process where we bring in driver projects for input. We have a committee that reviews the updates and the new standards, and we ensure that it covers the needs of, I think, at least 2 driver projects. So there is a whole process involved, and I see that the link was shared. And so as each standard comes out, we also have an open commenting period. As we had mentioned when we were going through the work streams, we have several deliverables right now that are open for comment. So we are always looking for feedback from the community when we build these standards and update them. Julia, I don't know if you have anything to add to that.
No, that sounds great.
Oh, sorry.
I think if
I checked that off by accident.
I think it was asking, yeah, what cloud platforms are implementing the WES standard, I think was the question. Yeah. So I'm going to try to answer this off the top of my head, but please reach out to us if you want, and we will put you in touch with the leads of the cloud workstream. But I know that, and Justina, jump in if I'm wrong, the Amazon Genomics platform, the AWS Genomics platform, is using the WES standard. I believe that Microsoft as well, I believe, uses the WES standard. Justina, do you know if there are others?
Unfortunately, I don't know the answer to this one. Oh, DNA Stack Elixir 2, I believe, is what was shared in chat. Thank you, Neerja.
Yes, thank you. But please reach out to us, and we'll put you in touch with David Glazer and co from Cloud Workstream, who can answer much more thoroughly.
We can give a few more minutes for any other questions. And of course, if you don't have any questions now, but maybe something comes up later, feel free to email us. We're happy to answer them as well post the webinar. All right. I think we can wrap it up just a few minutes early. Julia, if you want to help close the webinar.
Yes. So thank you all so much for joining and for asking questions and participating in our polls. If you have any additional questions or you want any sort of follow-up, please contact Secretariat at GA4GH.org. We will be sharing these slides after the webinar, so don't worry if you didn't get a chance to screenshot anything. And we appreciate your interest here in GA4GH, and we hope to see you on another call soon.